Hi guys, so this is about cell physiology, the material coming from the textbook Guyton and Hall, chapter 1 to 5. That's a lot of materials, so if you want to read more detail about those, you can read the textbook. But in terms of the exam, that will only coming from these PowerPoint slides, our lectures. You can get this textbook from our Loma Linda library, and that's free. You can read it through. You can even download the PDF file from the library, so you don't need to buy the book. This is the same book that we use for the medical school students. So it's there's a certain depth to it, but don't worry. We are not going to talk all those materials. We will only focus on the important essential portion. Cell membrane. It's also called a plasma membrane. And this plasma membrane, the major component is phospholipid. It's double layer phospholipid, and the other component is the cholesterol. So in the quiz question, what's the components in the cell membrane? The major one is the phospholipid, and another one is cholesterol. Cholesterol's function is to stabilize the cell membrane because this cell membrane is very liquidic, that they can move around, but cholesterol can enhance that stabilization. And with that, we separate intracellular and extracellular space. So everything inside of that plasma membrane is called intracellular space. And that space is also, the fluid is called cytosol. And the fluid plus the materials, organelles, etc., proteins, is called cytoplasm. So that's the intracellular. And we also have extracellular space. So this extracellular space basically include the major one is the plasma. Plasma means the blood fluid, as well as the interstitial fluid. So those are the extracellular space. Now, with this membrane, in addition to the lipid, we also have protein. Protein has several functions. Here you can see the cartoon, like a simplified image or picture showing you this membrane. So here you can see that here we have the phospholipid and transmembrane. So these are all transmembrane proteins. And you can see that one major function of the transmembrane protein is for transport. Here we didn't show you the channel, but later you will see a channel that substance can go through it. It's very important that these channels allow ion to go through it, allow glucose to go through it. It's because that without this channel or transporter, water-soluble substance cannot go through it. And the cells need glucose, because glucose provides the energy for cells to survive, to live. So that's very important. And this membrane protein can serve as a receiver. As you can see here, ligand binds on it, which can cause the signal transduction into the cell, can serve as a channel. Water-soluble substance can go through it. These channels can be gated, can be ligand gated or voltage gated. We will talk about those. And the carrier is a picture, it's not showing here, but very much like a transporter that will bind the molecule and move it to the other side. It can be either way, from the intracellular to extracellular or from extracellular to intracellular. It can serve as an enzyme, so this is very important that this enzyme can be used, is needed for metabolism, that we need to break down large molecule to be smaller molecule, or we can convert the inactive proteins to become an active proteins. It can serve as an anchor, so this is important to anchor what? This is usually proteins to anchor either the extracellular matrix, so those large molecules that filled in the extracellular space, or it can serve as the anchor for the intracellular cytoskeleton. There are 3 types of cytoskeleton. We will talk about those. And it can also serve as recognition proteins that can be used to label cells. For this facilitated diffusion, a typical one is a channel. This is also a protein. This is a transmembrane protein. A typical protein is is begin by the linear sequence of amino acids. So the primary structure is a linear, is like string, and this protein need to be folded. This original form need to be folded. So you can see that it has to be either form this alpha helix and then fold it together to form this channel, and anchor that channel across the membrane. So it has 2 opens, one end open to the extracellular space, the other end open to the intracellular space. So this is a very simple type of the channel, and that channel allow water-soluble substance to move across. This channel can be gated. There are 2 ways to gate it. One is called voltage-gated channels. So only opens or close when the voltage, the membrane potential is correct, is the right one. Then it will open or close. The other one is called ligand-gated. Ligand-gated is controlled by the chemical. So they have the receptor of certain chemicals. When chemical binds on it, it causes it to open or close. Or it's non-gated. If it's non-gated, it's open all the time, and this is also sometimes called a leakage channel, that things can diffuse through this channel and without specific control. So those are the diffusion. As for the active transport, this one requires an ATP. It moves the substance against concentration gradient. The typical one is sodium-potassium ATPase. It pumps 3 sodiums out of the cell and pumps 2 potassium in the cell. This is very typical one, and this one requires ATP. So this is this pump, sodium-potassium ATPase. This one requires ATP, and it pumps 3 sodiums out, 2 potassium in. So the cell need to have glucose, need to have the fuel. It's because that it needs to have the ATP to maintain the function of this sodium-potassium ATPase. Now with this sodium-potassium ATPase, it leads to two consequences. The first one is the ion concentration difference between intracellular space and extracellular space. Here we have constantly pumping 2 potassium in and 3 sodiums out. It will lead to an increased concentration of potassium in the cell and the 3 sodiums and the sodium out of in the extracellular space. So it leads to that. The first consequence is the concentration difference. One thing you need to memorize is that in the cell, everything is low except potassium. Okay. So potassium is high intracellularly. So that's the first thing. That's one thing you need to memorize. You will see it in the exam. Which ion has higher concentration inside of cell than outside of cell? The answer is potassium. So that's first consequence. The second consequence is that when it pumps positive charge ion, and but not the same amount. So 2 in, 3 out. So it pumps more positive out of the cell. And it will lead to the consequence that the intracellularly it has less positive charge ion than the extracellular, and it will make the intracellular to be appeared to be more negative compared to the extracellular. And that built the foundation of the membrane potential, because that uneven positive charge ion movement, 2 inside, 3 out of the cell, will make the intracellular to be more negative. So the second consequence is membrane potential to be negative. Because we have this sodium potassium ATPase almost, I don't recall one cell which doesn't have it. Maybe I missed some, but almost all cells have the sodium potassium ATPase, and that will cause this consequence that all cells have membrane potential. All cells has a negative. It's around negative 70 in the most of cells, negative 70 millivoltage MV in the cells, then the outside. So that's the consequence. So two consequences: the ion concentration difference, and the second one is a negative membrane potential. So this negative membrane potential is driven by the major cause is the sodium potassium ATPase. It's also caused it's also contributed by the potassium leakage. It's because that we constantly pump potassium in, right? And in the end, that potassium will gonna overflow. So you must have some leakage in the cell membrane to make it like balanced, because you keep pumping it actually all the time, but you don't see the potassium like a burst. It's because that we have this leakage channel. So those are the two contributing factor to maintain a stable membrane potential and as well as stable potassium concentration inside of the cell. Those two contributing factors are sodium potassium ATPase as well as the potassium leakage channel. So this slide summarizes what we have to provide the material transport across the membrane. We have the simple diffusion across the lipid bilayer, and that's lipid soluble substance, which include O2, CO2. So here we have a lot of quiz question, like what can be moved across the cell membrane? O2, CO2. Okay, those gases, they don't need channel. They are lipid soluble. They are not very good water soluble substance. You know that, right? Gas cannot. Gas can be dissolved in water, but not a lot of amount. So we do have a little bit amount of gas dissolved in the water. But they are very good lipid soluble, so they move across the cell membrane. The next thing is the steroids. Steroids what? Steroids hormone. Our body generates several steroids hormone. All these sex hormones are steroids hormone. Adrenocortical hormones are steroids hormone. So these steroids hormones are made by lipid, made by cholesterol. So they are lipid soluble. They can move across the cell membrane. So when the cortisol, for example, cortisol, they don't bind on anything on the cell membrane. They see the wall, they see the cell wall as nothing. They just move across to it. They are like ghosts, penetrate the wall, go into places. So they will move across the cell membrane freely, easily. So those are. And what else? Vitamin A, D, K, E. These are lipid soluble, so they diffuse across cell membrane. Another one is water. Water can move across the cell membrane, and that is driven by, it's also simple diffusion, but it's also a specific term for it. It's called osmosis. So basically, water movements from highly condensed area, from the low condensed area to high condensed area, because in high condensed area it has less water, so water will move from very watery area to less watery area. Less watery area is usually the place where we have highly condensed particle in it. So water can also move across the cell membrane without channel. And the other one is facilitated diffusion. This one is provided by the channel. And all these are passive transport components. We also have the active transport component. Then the major pump is the sodium-potassium pump. So that's the membrane. The key here is that the major components of the membrane is the phospholipid, cholesterol. So that's the lipid. We also have the protein. These transmembrane proteins serve many functions, including the channels, including the enzymes, anchors, etc., receiver. And these proteins also provide the facilitative diffusion, as we mentioned here, as well as to serve as a pump. Back to this text here. So that's the carbohydrates. They serve in the extracellular space, in the extracellular side of the cell membrane as a sugar code. And this is also very, very important knowledge that you have, that you need to have, is that, for example, if our blood, have a different level of glucose, for example, that patients with diabetes, those glucose will affect the cell by altering their glucose binding to the cell membrane, and that actually can change the membrane function, change the protein function, and eventually change the cell's function. So that's why the patients with diabetes may develop a lot of vessel disease. It's because that this glycolization alter the function of the endothelium cells, and that alter the vessel wall structure, vessel wall function, and to cause vessel disease. All right, so that's the membrane. The next stop is the organelle. So we will choose the major organelles to kind of give you an overview and to lay the foundation for the future course. These organelles are distinguished into two groups. One is called non-membranous organelle. This one doesn't have the membrane. The major two are the cytoskeleton and the ribosome. The other group is called membranous organelle. This one do have the membrane. A lot of other very typical organelles are membranous organelle. It's also called membrane-bound organelle. So they are nucleus, ER, endoplasmic reticulum, Golgi apparatus, lysosome, microtubule, etc. Here is a picture showing you that it's not empty space when there's no organelle. It's actually filled with this cytoskeleton in the cells. So microfilament is composed of actin. This is dynamic, composed, discomposed. Intermediate filaments is more durable, composed of several types actually. Collagen is just one of it. Microtubule is the largest one, hollow tube, and it's composed of tubulin. Microtubule has several interesting functions that you need to keep in mind. Microtubule is the structure for cilia. They can move like a whip, so cell can swim. It's also the structure for flagella. Very similar like cilia. They can move, they can swing, and make the cells to move. Alright, so for those who don't move that fast or swim in the fluid, microtubule is also important for cell division. So this provide the like a string for chromatin, for sister chromatins to be separated. So they serve as this spindle apparatus during the cell division. So that is also very important, microtubule. So they have to be divided and assembled very rapidly in order to provide that kind of dynamic change. Tumor has cell division very rapidly, and one type of tumor treatment or cancer treatment is to inhibit the microtubule assembly and the disassembly, so that can prevent the cell division. It's also important for exon transport. So this is neuronal cell. Neuronal cell has a very long axon. That materials need to move from cell body into the axon terminal they cannot just depend on the diffusion. For example, you have the neuron soma in the eye, and the axon project into the back of your head, your brain. And if it's just by diffusion, it will take forever from the proteins to move from the eye into the back of your head. So it has to be transported in some way. And that transportation inside of the neuron is by axon transport. And that axon transport is achieved by this cytoskeleton called microtubule. So those are the important cytoskeletons. You need to know the components acting for the microfilament, tubulin for the microtubule. You also need to know that the major function of that microtubule in to serve this cellular function inside of the cell. Those are also very typical quiz questions that you will see from this type of class. All right, so another non-membranous organelle is the ribosome. Ribosome, the function of ribosome is to conduct the translation. So it will convert RNA into protein. Ribosome can be freely diffused in the cytoplasm, or it can be fixed. The ribosome binds on the endoplasmic reticulum. So this is ribosome. Ribosome has two components. When they are not active, these two components are separated. And when they are active, these two components close together and provide that space, like and RNA will go through it, and it will generate protein. So the function of ribosome is for the translation is to generate protein. So here is the gene expression from DNA to RNA to protein. So DNA in the nucleus through transcription to produce the messenger RNA, mRNA. Then the mRNA flow out of the nucleus into the cytoplasm, and through the ribosome, it generates protein. Ribosome can be freely diffused in the cytoplasm, and when it generates protein, it will produce it directly inject them into the cell. You can also find the ribosome attached to the endoplasmic reticulum. So this is also called rough ER in comparison to the smooth ER. Smooth ER is the endoplasmic reticulum without ribosome. And this ribosome will also do the translation to produce protein. When they produce protein, this protein will be injected into the endoplasmic reticulum, where they will be further processed. They will form the vesicles, move to the Golgi apparatus, and then it will be either transported to other organelles, or it will be delivered into the extracellular space. Example of the membrane bound. Here is the ribosome attached to the rough ER. The protein will injected not into the cell cytosol, but injected into the ER. And inside of ER, it will be the first structure of the protein is the linear structure. So it's a string. It's a connection. It's a polymer of the amino acids, and it need to be folded. And it's folded, then it will pack it in the vesicles. This vesicle will be pinched out. Then it will flow to the next stop. Usually the next stop is the Golgi apparatus. And over there it will be further processed, further folded, further decorated to become like a functional protein. So those two are the non-membranous organelle. Cell skeleton, 3 type, ribosome, 2 type, free and fixed. And you need to know their function. Now the next group is the membranous organelle. We have the nucleus. This is the double membrane organelle. Endoplasmic reticulum, we kind of mentioned about it already, is the place where protein is injected into and processed, and the protein will be folded and packed in the vesicles. Golgi apparatus, lysosome, peroxisome, mitochondria. One thing you need to know is that these membranous organelles, 2 of them has double membrane, nucleus and mitochondria. These 2 has double membrane. So the function of the nucleus is that it contains chromosome. Chromosome is place that contains the DNA and our gene. This double membrane together form is also called nuclear envelope, and it also has nuclear pores that RNA can freely diffuse across through these pores from the intranuclear to extranuclear, and to be processed by the ribosome. So nucleus contain the chromosome. In our body we have 23 pairs. Pairs means that for these somatic cells, not the gamete cell, not sex, not the sperm and eggs, but in the somatic cell we have 23 pairs. In the gamete cells we have the only 23, not pair, 23 chromosome. In the somatic cells, our body cells, we have 23 pairs, one mom, one from dad, and 22 autosomes and 2 sex hormones. And this chromosome is combined, include DNA and protein. Okay, it's not just DNA, it's DNA and protein. DNA contains is a nucleotide and the polymer of nucleotides, and the DNA folded using the protein. This protein is called cystone, and folded condensed form to form this chromosome. And it is direct transcribed from the DNA code to RNA code. This RNA sequence is called the primary script, and then it needs to be further split into the matured RNA. This split is called it can have alternative splicing. So let's talk about that. This is due to the exon and the introns in this primary script. So here is the DNA and transcribed to the RNA. So you can see this primary RNA, it contains segments in exon and intron, exon and intron. Introns are not used, they will be removed. Intron is also called junk DNA. For a long time, we don't really know what's the purpose of that group, of that segment of the DNA and RNA. But later we found that they have several regulatory functions into the gene expression. But at this point, you just think that these introns are not used. We only need exon. So exon then binds together then it will form the matured RNA. This process can lead to different variety of RNA sequence. For example, that here is a primer script. We have exon 1, intron 1, exon 2, intron 2, exon 3, for example. So the same sequence can be used to generate different protein by alternative splicing. So intron are all removed. Now we put these exons together. When we put them together, it can lead to either we just accidentally don't use not accidentally, this is regulated in the cell. Then we have the matured RNA include exon 1, exon 2, but not 3. Or we can have exon 1, exon 3, but not 2. Or we can have exon 1, 2, 3. So just by that selection, we can generate alternative messenger RNA and lead to the alternative protein. So that's the alternative splicing. It's a selection of the exons and to build the variety of different proteins from the same sequence in the DNA. This one become very important in several disease development. For example, in the Alzheimer's disease, that everyone has the proteins called tau expressed from this large DNA sequence, but just by the alternative splicing, people can generate different species of these tau proteins, and some tau proteins have high tendency to form this neurofibril tangle, and that is one pathological hallmark of Alzheimer's disease. And so the ratio of the pathologic proteins versus the non-pathologic protein can define the progression of the disease. And so the alternative splicing is an important mechanism that our body can generate a little bit variety of these proteins from the same like a sequence of the DNA. What? Yeah. All right. So and this ribosome attached to the ER will translate the RNA into protein. The first structure of the protein is a linear structure, and it is like a string of the bits. It's not functional. It has to be folded, and to form the protein, they will be packed in the vesicles. These proteins are very carefully separated or packaged, so they are not easily flow into the cytoplasm. Different protein has different function. Some of this protein can be digestive protein, so we don't want them to see things in the cell, otherwise they will start to digest the cells. We don't want that, so we pack it, and then we will move it. We will guide these vesicles into the place that it's supposed to be. Smooth ER, the major function of a smooth ER is because this smooth ER is a membrane-bound organelle. It contains membrane. This membrane are lipid. So this is a place to form a lot of lipid or collect the lipid. If the cell receive the lipid, they can use this smooth ER to manufacture more of these lipid products and attach it into the membrane. So this place to produce the lipid for the vesicles. Then these vesicles will then travel to the next stop. It's usually a Golgi apparatus. Golgi apparatus will first kind of engulf it, and the inside of this process, they will further decorate. It's called post-translational process to decorate these proteins and to fold it, and then it will move it into the vesicles. This is secretory vesicles. It will then dump this protein into the extracellular space. Something like, for example, insulin. Insulin will be released into the blood and to help us to transport glucose. So that's how, for example, insulin produced from the DNA transcription to RNA. RNA translate to the insulin protein. Insulin protein need to be folded and stored in these vesicles through the Golgi and waiting here. When we eat, insulin will be released to help us to store this excess glucose. When they release into the extracellular space, it's called exocytosis. So this is the vesicle. The vesicle has the same material as the other cell membrane. It has the phospholipid. Vesicles can move out, can serve as this package to export them into the extracellular space. It's called exocytosis, or it can engulf the extracellular space material into the cells. It's called endocytosis. So just showing you that the vesicles from the Golgi can go into the lysosome, and the lysosome is the organelle to digest material. Our cells, for example, the macrophage can engulf bacteria, and it will, when it engulfed, this will form this endocytosis. This endocytosis vesicle will merge with the lysosome. Lysosome contains the digestive enzyme. Then digestive enzyme will start to hydrolyze to break apart these engulfed bacteria. And basically, bacteria has a cell membrane. We will break it apart, and we can either use this material for our body to use. They basically is a building bricks for cells, and we can probably collect some protein from there too, or we can dump the what we don't need into the extracellular again, and we can basically just remove it from our body. So lysosome contains the digestive enzyme. It's an intracellular digestive system. It contains the enzyme to hydrolyze, to break apart this large molecule into the amino acids, glycogen or carbohydrate, polysaccharides to the glucose, lipids to the fatty acid, and glycerol. One kind of like give you a synchronized structure or process of how nucleus, ER, Golgi, and lysosome work together to either move material into the extracellular space, or move material digest it and use it for our body's use. Another important organelle is the mitochondria. Mitochondria also has double layer. It also has DNA, so it's very much like another nucleus that has the double layer, and it does have the DNA. So mitochondria has two layers. One is called outer membrane, the other one is called inner membrane. The space between is called intermembrane space, and the inside of the inner membrane is called matrix, and inside of this matrix contains enzyme for the oxidative phosphorylation, and the major function of the mitochondria is to produce ATP, and that's what we need for a lot of body of cells function. To produce ATP is basically it's a phosphorylation process. It phosphorylates ADP. ADP has diphosphate, so it has two phosphate, and we add one to it to become triphosphate. So it will be a phosphorylation to form the ATP. In the mitochondria, this phosphorylation is done through the process called oxidative phosphorylation. So oxygen is required, and by doing so, it generates a lot of ATP. So it's very powerful organelles to produce the ATP that our body needs. It's a place that we can convert the energy from our food to the energy that our cells need, from the glucose to ATP. So the process to produce that ATP is through the oxidative phosphorylation. So this one is done through a group of proteins located in the inner membrane. So here is the inner membrane. This group of the proteins in the inner membrane is called electron transport chain. So they basically pass through the electron, and this electron will eventually be reduced by the oxygen to form water. So that's why we need oxygen. So let's see what it does. What's the major goal of this electron transport chain? So basically, that the purpose is to produce ATP, is to cause the oxidative phosphorylation to produce ATP. And then how does it do it? So it basically, what it does is that it will use two sources, NADH and FADH2. And the NADH and FADH2 donates their hydrogen. It contains the hydrogen here, and the hydrogen is then pushed into the intermembrane space. So in this intermembrane space, it accumulates a lot of hydrogen, and that hydrogen concentration will provide that concentration gradient to push through this ATP synthesis, this channel. When you go through it, it activates it, and that activation will convert ADP to ATP. So that's basically how it does it. We have the NADH and FADH2 to donate the hydrogen, the proton, and those are accumulated in the intermembrane space. Then that high concentration pushed through is like a windmill, pushed through this ATP synthesis, and that process, that kind of concentration gradient hydrogen movement will convert the ADP to ATP. So that's basically how it was done. And through this process, electron is transported through these proteins, and that electron is received by the oxygen and to form water. So oxygen is needed. So the thing you need to memorize is that the molecule to provide that, to donate the hydrogen here, are two major molecules, that is the NADH and the FADH2. And you also need to know that these oxidative phosphorylation through this electron transport chain take place in where? In the inner membrane, on the inner membrane of the mitochondria. These are the protein in the inner membrane. So that's why in the inner membrane you have seen a lot of this folding. That's because it needs to generate more space for this process to be done. And oxygen is needed. So that's the three major things: NADH, FADH2, inner membrane, and oxygen is needed. Also, another thing is that hydrogen concentration is high, or hydrogen is accumulated. Hydrogen ion are accumulated where? In the intermembrane space. So that's also something you need to keep in mind to memorize. You will see this in the quiz though. So that's why we need oxygen. We need to breathe. The matrix. So here is a citric acid cycle or Krebs cycle to produce that NADH. And that basically is the process to convert glucose into NADH and FADH2. Here we don't see the glucose here. Glucose first need to be processed through the glycolysis to produce the pyruvate. Pyruvate then need to be converted into the acetyl-CoA, and then acetyl-CoA, as well as and then it will go through this cycle to produce a lot of NADH and FADH2, and that NADH and FADH2 will then be used in the electron transport chain to donate its hydrogen, then to provide this oxidative phosphorylation. So citric acid cycle is done in the matrix. So that's a quiz question you will see. Where? In the matrix of mitochondria. Here in this process, it produces CO2, and that's what we need to breathe out. Just keep in mind that the location where we produce CO2 is different from the location where we need oxygen. The region we produce CO2 is in the matrix, in the inner cavity of the mitochondria. The place we need O2 is in the inner membrane, where we have the electron transport chain to conduct the oxidative phosphorylation. That's where we need the oxygen. So the production of CO2 and the need of oxygen is actually in two different processes. So citric acid cycle is important to provide the NADH and the FADH2. This one is done in the matrix of the mitochondria. So here you can see mitochondria is important, that it basically converts this food, energy source, glucose, we need to eat, carbohydrates, we need to eat, protein, or we need to eat lipid, in order to convert them. The place to convert them into the ATP is in the mitochondria. It will go through it, produce the NADH and FADH2 in the citric acid cycle. Glucose first to convert into the pyruvate into the acetyl-CoA, and through that cycle will produce NADH and FADH2. In the electron transport chain, it will produce the ATP. summarize it. That's all you need to know from the cell. Okay, and then we will move on to the next step, which is the muscle. And with this one in hand, you will know that how we convert glucose into the energy, and what's the structure in the cell. We talk about the cytoskeleton, 3 types of cytoskeleton. In the muscle cells, we will learn that one of that cytoskeleton, the actin filament, is an important component for muscle to conduct this muscle contraction. So I will see you next time.